introducing us, and I'll explain. But first of all, I've never had the pleasure of meeting you in real life or virtually. I'm Rachel Wimberly. I'm the Director of Adult Programs and Arts and Culture at Shalom Austin in Austin, Texas, in case you're joining us from elsewhere. And then my amazing colleague, I like to like Vanna White to the left, but she may not be the left on your Zoom, is Annie Skelton, who is our program coordinator. And we are here going to introduce Tom, and Annie's actually the one that's done the work and set this up and invited Tom. But we are here managing things. So if you are having a Zoom issue or something happens, you can always chat one to everyone, or there's a little chat bar at the very bottom that looks like a little chat bubble or like a talking bubble from a comic book. You can always chat Annie or I individually and ask a question or ask for help. So I'm going to introduce Tom at the end of this sort of intro. So a few housekeeping things. Once we get started, we will be muting everybody but Tom so that he can speak. And there's just no, even if you're not talking, sometimes there's just like, I have a 7-month-old baby that could cry. So there can be some noise pollution. So we'll just mute everyone so we can hear Tom really clearly. He will speak for about 35 minutes. And then he'll answer questions. So while he is speaking, you can, if you have a question, you can either jot it down, or again, if you want to use that chat function at the bottom of Zoom with the little like talking bubble, you can put the question in the chat, and we won't interrupt him while he's speaking, but we'll sort of keep them all there, and then we will I'll ask Tom at the end, or he'll read them. So if you have questions and you don't want to forget, just pop them right in that chat bubble during the presentation, and we'll address them at the end of the program. As I mentioned, for some of you folks coming in, we are going to record this program for those who maybe can't stay the entire time or aren't able to make it today. That way we can share it. And I will send an email once the video is up on the Shalom Austin YouTube page to everybody in this room. So you don't really even have to worry about going and looking at it, looking for it. Tom, you have a really illustrious and long introduction, so I might skim parts of it. So don't take, I mean, he's got way more to say than I can say about him. But today's presentation is about the work he did as a research analyst in the biological sciences section at the Lunar Receiving Laboratory during the Project Apollo from 1969 to 1972. There he examined the lunar soil and various animal species to look for pathogens that might be harmful to the biosphere. After the project, he worked as a research associate in genetics, infectious diseases, and radiology at the University of Texas Medical Branch at Galveston, and then gradually moved into scientific medical media as manager of medical photography at Texas Tech University Health Sciences Center in Lubbock, which really means that we could probably have Tom come talk about lots of various parts of his career, because there's so many in that intro that I also want to hear about. But without further ado, I'm going to hand over the floor, the Zoom floor, to Tom. I also meant to mention, as he's about to get started again, when we will mute ourselves, Tom's going to share his screen. So you can keep your video up. He won't really see that much, and we won't really see each other because he's got a really great PowerPoint with really cool images. But if you want to, like if you have any Wi-Fi problems, or you just want to, mute your screen or turn off your video, and then when we come back for Q&A, we can all turn our screens back on. You don't necessarily have to keep that open. We won't see you, so it's up to you. So without further ado, I'm handing it over to Tom. The floor is yours.
Okay, my voice is not working very well these days, but I'll try. Thank you very much, Rachel. Welcome, everyone. I want to thank Shalom Austin, their adult education department here in Austin, Texas, where I'm based as well, for giving me an opportunity to share my Project Apollo experience with its membership. I also want to thank other guests who are in attendance today. As Rachel mentioned, I'll be using I'll be using a PowerPoint presentation, but this is the same PowerPoint presentation that I used at the Wings over the Rockies Air and Space Museum in Denver, Colorado, for the Apollo 11 50th anniversary celebration last year in July 2019. So with no further ado, as Rachel said as well, I'm going to bring up a PowerPoint. It's rather lengthy, but I think it should detail my work experience at the Lunar Receiving Laboratory, and I hope you will enjoy it. So let's see if I can share the screen. Everyone see that okay? Rachel, okay?
Yes, you're good.
Okay,
Looks great.
Okay. Okay, just a moment. Let me, okay. So by the way, my memory these days, I'm 73, so actually I'm one of the younger out of the group. Most of the people I worked with back then, 50 years ago, were unfortunately deceased. So I started very young, working with the team at the age of 22. I had just turned 22. So my memory's not the best, so I will probably be referring to this slide presentation and reading from it most of the time as I talk to you. So here we go.
Tom, real quick, do you want to make it just that slide? Because right now we can see the.
Oh, I'm sorry. Yeah, you're right, Rachel. Excuse me. Sorry.
No, you're totally fine.
Yeah, got it. I got it?
Perfect.
Okay, great. So I thought I would begin with a diary entry from my grandmother, who I miss. On July 20th, 1969, she made this diary entry, and she says, Jean and Joe, my parents, came up and we watched the men on the moon. I thought that was great because we always think of man in the moon. She had mentioned we watched the men on the moon. She said, didn't I go to church? Sorry, I didn't. So I wanted to begin this way. She doesn't mention me, but I was with my parents and my grandmother on the 20th and watched Neil Armstrong and Buzz Aldrin walk on the moon. I had graduated from college just 2 months earlier, and little did I realize at that time that I would be working as a research analyst at the Lunar Receiving Laboratory. So the title of the presentation is 50 years ago at the Lunar Receiving Laboratory, Project Apollo. By the way, this image is of the Apollo 12 core tube on the surface of the moon. The top left photograph shows me in the very top left of the image, and my boss, team leader, Landry Young, who I believe is deceased. I've lost contact with him over the years, but I believe he is gone. He was the team leader. And in this image, you see us working in the containment cabinets that isolated us from the lunar soil and vice versa. Landrum injects a quail as I assist him, a suspension of saline and lunar soil brought back from the moon by the Apollo 11 astronauts. That solution was prepared for the injection. Again, I've mentioned, notice the containment cabinets. We work this way. I got in on the tail end of 11, but we worked this way for 11, 12, 14, and even 15, 16, and 17, we still handled the lunar soil within a containment cabinet so we didn't contaminate the soil at that time. For 11, 12, and 14, we were worried about back contamination, the soil infecting us, our biosphere. The photograph bottom right, I'm seen in front of the Lunar Receiving Laboratory, building 37. which I visited in 2016. So it had been about 44 years since I returned to the lab. And as I mentioned to Rachel yesterday, time does a number, 50 years. I joined the Biological Sciences Research Team near the end of the Apollo 11 process, which I think is interesting because one of the team members breached the containment protocol and was then quarantined with the astronauts. I certainly won't mention the analyst's name. I have been working as a research analyst at the MD Anderson Cancer Center in Houston, just right out of college, first job out of college, but always looking for the grass is greener, looking for another position. This NASA photograph, again, shows my team leader, Landrum Young, working with the mice, just one of the many species we worked with, in the containment cabinet during Apollo 11. You'll notice the double barrier, the isolator, which I'll move my cursor, the isolator keeping the mice protected, the germ-free mice, and then the containment cabinet. Germ-free mice lack all microorganisms, and they are housed in tightly controlled and monitored isolators to prevent contamination. They are microbiologically sterile. No living organisms can be cultured in germ-free mouse specimens. So we worked with this kind of mouse. The images below show examples of biological or containment cabinets. I spent a great deal of time working in these cabinets during missions 11. This quarantine approach was to help prevent forward contamination, the transfer of life and other forms of contamination from Earth to another celestial body, in this case, the lunar rocks and soil. And more importantly, back contamination, which I have mentioned, the introduction of extraterrestrial organisms and other forms of contamination into Earth's biosphere. So we have a nice schematic to the left and a NASA photograph of some of the containment cabinets at the lab. Although the formal quarantine for the crew, spacecraft and lunar samples was over after Apollo 14, procedures for handling lunar material and protecting it from contamination remain in effect for the Apollo 15, 16, and 17 missions. I remember the first day, or actually night. I began on an evening shift at the laboratory. When I joined the team, they were in the midst of processing the lunar soil core sample by the Apollo 11 astronauts. That first night was as surreal as it gets, and I do indeed remember that. I entered a change room and put on a bunny suit similar to a surgical uniform, walked through a UV wash, dry shower, and then ended up at the beginning of a long corridor, which you can see here. So I've moved my cursor on the image. There were doors running to the left and right all the way to the end with autoclaves, sterilization units interspersed on the walls. And you can see these units here. I believe these NASA were actually pre-mission promo pictures that were taken by NASA before the missions even began. There were doors running to the left and right all the way to the end with autoclaves interspersed on the walls. The laboratory where I would be working was at the very end. The team leader, Landrum, entered a combination code to open the door, and then we entered. When I entered, the first things to catch my eye were the large containment cabinets, metallic, glassed, see-through working areas with glove ports. I had never seen anything like this before, except for a hooded work area in a microbiology laboratory. I would spend the next several months with my arms inserted into those glove ports, working with the lunar soil from missions 11, 12, and 14 in various animal models, species. The team leader and other analysts would be at my side as we worked the samples for analyses. You notice the image to the right, all of the gloves are extended outward because of positive pressure. As we worked with the lunar soil, we were working with negative pressure, gloves inward, to prevent back contamination. That first night on the job of a lifetime, which I truly mean, left me a bit numb. After 8 hours working in a surreal setting, bunny suits, UV wash, containment cabinets, sterilization units, the tension of having to be very careful not to breach the containment protocol, and of course, the lunar soil sample on the other side of the glass. This is a great schematic, came out in Life magazine, that shows the layout of the Lunar Receiving Laboratory. The red circle are actually the two labs that house the animal model, various species, particularly the mice. I should have circled this lab right here, but this is the lab where I did most of my work, where I'm moving the cursor. I was in that particular room the majority of the time for 3 years, and which I think very isolated, by the way. We were very compartmentalized and very busy. We didn't have time to nose around. You'll notice, which I think is interesting, when I look at this schematic, you'll notice the astronaut trailer here and the Apollo capsule here. So these two pieces of hardware are very, very close to me, but I never ever saw them because we were so compartmentalized and so busy. And maybe even probably, if we were to try, maybe prevented from going into those areas. But I think that's interesting, very, very close to the hardware, but I never saw the hardware at that time. Too busy doing my job. As I've mentioned, the lunar samples were tested within biological containment cabinets. These cabinets were gas-tight enclosures through which all manipulations were performed using neoprene gloves. Air or nitrogen entered the cabinets through absolute biological filters and was filtered again before being vented to the outside. All material entering the cabinets was sterilized. The cabinets were operated at a pressure negative with respect to the laboratory to ensure that any leak that developed into the cabinets rather than into the laboratory. There was also a secondary biological barrier. The rooms, the labs, in which the cabinets were housed were also maintained at a pressure negative with respect to the adjacent corridors. This guaranteed that any escaping lunar material would be contained. This secondary biological barrier, which surrounded the sample laboratory, included facility systems and operational procedures. Tight building construction was used and all penetrations were sealed. All solid materials, including waste, clothing, and trash, were sterilized. The sample laboratory area received supplies during quarantine operations through ultraviolet lighted UV airlocks. And of course, I'm taking a lot of this information from NASA literature manuals. So of course, I need to credit NASA for a lot of this text. I remember these airlocks very well since I had to take a UV shower each time I entered and also exited the building. There were three main elements as part of the biological protocol: crew, microbiology. I had a very good friend who worked in that area, who I will mention later again. In vitro attempts to culture microorganisms from the lunar sample, and the direct challenge of the lunar sample in biological systems. I was involved in the third element. The group of hosts involved higher and lower vertebrates, invertebrates, unicellular organisms, and plants. I can briefly go over which I think are interesting. The existence of hazardous replicating microorganisms on the moon would be assumed. Biological containment requirements should be based on the most stringent means used for containment of infectious terrestrial agents. The sterilization requirements should be based on methods needed for the destruction of the most resistant terrestrial forms. Hazard detection procedures should be based on an alteration of the ecology and classical pathogenicity. The extent of the biological test protocol would be limited to facilities approved by the Congress, to well-defined systems, and to biological systems of known ecological importance. Maybe we should apply these guidelines today, huh? Towards COVID-19, anyway. These guidelines provided the basis for the lunar quarantine program. Although the probability that life existed on the moon was extremely low, the risk was sufficiently high that a quarantine program was justified. There were a lot of cons out there too, but thankfully the pros outweighed the cons, which I think was smart. The term hazard had to be defined before a method of detection could be developed. Procedures were limited to those capable of detecting an agent that would exhibit classical pathogenicity to some terrestrial life form, or that could establish itself in a terrestrial environment and thereby alter the ecology. This guideline limited the search to the detection of replicating microorganisms. As it related to my responsibilities in the biological sciences section, the methods used for the detection of replicating microorganisms that could cause disease or establish and replicate themselves in some terrestrial environment or organism were as follows. Lunar from the core samples. You see this great picture of the core tube, Apollo 12 core tube, were prepared for injections and administered. The various animal species or models were sampled at various time cycles, bled or exsanguinated for blood analyses and chemistries, dissected for light and yes, even electron microscopy of various tissues to look for changes in morphology, and cultured for microbial procedures. Our main focus when we received the Apollo 12 sample was to prepare saline lunar soil suspensions for injection of groups of mice and other species in staggered fashion for incubation and processing. At various time intervals, the mice would be bled for blood analyses and dissected for tissue sampling. Keep in mind, all of these precise and tedious procedures were done through bulky gloves with restricted movement and viewing limitations, in the sense that you had to be careful that you didn't bang your head on the glass window of the containment cabinet. It was indeed grueling and tiring process. And again, we had to be careful that we didn't pinprick our gloves, which would have shut down the entire system. As I have mentioned previously, an analyst in our area did indeed do this during the Apollo 11 procedures and was quarantined with Neil, Buzz, and Michael. Great photograph. This work environment gave me a greater appreciation of the astronauts' work environment when they were on the moon working with procedures in their bulky suits and gloves. Isn't this is just an incredible photograph? I'm also a photographer, by the way, these days, and have enhanced and edited a lot of the Apollo images. It's just a wonderful photograph. Alan Bean, by the way, Pete Conrad took this photo of Alan, Apollo 12 mission. If I'm correct, and I'm trying my best to be correct, after 50 years, our prepared sample came from this Apollo 12 core tube, sample 12026. It was collected in drive tube 1 near the lunar module at the end of the first EVA period on the northeast edge of Surveyor Crater. The core was 19.3 centimeters long and contained 106.6 grams of soil. 3 small samples were taken from near the top, middle, and bottom of the core for gas analyses. Then the core was dissected and split longitudinally. The split was divided into 3 samples, top, middle, and lower thirds. Each sample was sieved, filtered, then recombined to form part of the bioprime sample, the sample used by us in the quarantine area. Again, credit to NASA literature manual. We then took this bioprime sample and made saline suspensions for our injections of the mice and other species. As I have mentioned, once the mice were subjected to the saline lunar soil suspension, it was an hourly process of macroscopic and microscopic observations with exsanguinations, dissections, to be able to do blood and tissue analyses. Over time, I perfected a technique of obtaining optimal blood levels with minimal hemolysis or damage to the red blood cells during the exsanguination process, and suggested to my boss, the team leader, Landry, that we publish a paper on this in a scientific journal. We did, and I'll never forget how we decided who should be on the author credits, or should be first, sorry, on the author credits. We flipped a coin, and he won. So his name precedes mine on the paper, which is perfectly okay. The paper was not only accepted for publication, but also won first place for best technical paper published in the scientific journal, Laboratory Animal Science. And I have a copy of the paper to the right. When I look at this again, it's pretty modest, a modest technical paper. But today, when I look at this, I like this bit of literary, if you will, scientific accomplishment now for its historic nature. What I think is very cool, if I may put it that way, in the legend, and I'll put my cursor over here, Incorporated Lunar Receiving Laboratory, NASA Manned Spacecraft Center. NASA and the program is referenced as a part of this paper, probably the main reason it won Best Technical Paper. I believe for the year 72 or 73, I can't remember exactly. But I'm proud of it now just for its historic nature. By observation of plant and animal diseases, it was determined that most terrestrial disease agents were capable of invading a host and causing evident disease symptoms within 21 days after exposure to the host. Most disease agents capable of causing epidemic or rapidly spreading diseases were sufficiently virulent to be transmitted in less than 21 days. So it was decided that a crew quarantine period of at least 21 days should be required after each Apollo mission. Intensive medical examinations of the flight crew members during quarantine determined if any medical problems existed as a result of exposure to lunar material. And this particular lab was very close to where I was working. And let me stop here and mention a member. I've already referred to his name, Rudy. Let me stop here and mention him. We became very good friends, and his enthusiasm for Project Apollo was infectious, no pun intended. He passed away several months ago, and I want to dedicate this presentation to him. This is a picture of Rudy to the right, fused with the moon. I miss him a great deal. I think he was a NASA type, and he remained at NASA all the way through, I believe, through the shuttle program, working in the medical laboratory. Our procedures in the biological sciences, should read section, followed similar guidelines with a repetitive approach to ensure release of the lunar samples to other investigative teams did not represent a hazard. The hourly process of macroscopic and microscopic observations with examinations to be able to do blood and tissue analyses was definitely a grind. We had to be punctual, right on, and deliver results at a fast pace to satisfy the concerns of the day. Is the moon safe? And we had to be careful not to contaminate the samples that were brought back. As tests were progressing for the Apollo 12 lunar soil, we also had to begin preparations for the return of the Apollo 13 samples. We set up procedures and modifications based on prior techniques with missions 11 and 12 and practiced the various approaches so they would become routine. Little did we realize that these projected routines would be broken. As usual, like the previous missions, we were anticipating the launch of Apollo 13, their journey to the surface of the moon to collect our sample, if you will, and their return to Earth. But the accident that happened to them along the way changed everything, and a doom and gloom cloud hung over the Lunar Receiving Laboratory. We had to be conscious of our ongoing procedures and tests with the mission 11 and 12 samples, but it was a difficult task to get our head around the fact that we might lose our Apollo 13 astronauts over a bunch of rocks. Of course, I remember this distinctly, and I walked into work those few days with nothing else on my mind except concern for Lovell, Hayes, and Swigert. To the right, you see the lifeboat that saved the Apollo 13 astronauts, Aquarius. Wonderful photograph. There was about a 4-month delay for Project Apollo because of the mission 13 problems. So we did some maneuvering as well, which meant I worked in other research activities. I remember walking into the medical laboratory area to utilize their electrophoresis equipment to do some enzyme work on mice that had been subjected to lunar soil. I began utilizing their equipment to establish enzyme baselines. I got pretty good at this too, and I ended up publishing my results in another scientific journal. Apollo 14 made it through the process. Everyone on pins and needles because of the Apollo 13 mishap, and we received our lunar soil sample. We were excited as ever as we opened the lid and witnessed the moon again, but a different part of this celestial body. The Apollo 12 sample was from the southeastern portion of the Ocean of Storms, and our new sample, Mission 14, was from the Fra Mauro Formation. Again, to the right, you see another wonderful photograph of Alan Shepard holding a core tube and his MET, modular equipment transport system here, cart. Great photographs. We essentially performed the same procedures, subjecting animal species to the lunar soil. The crews of Apollo 11, 12, 14 experienced no health problems as a result of their exposure to lunar material. The test species, plant and animal, which were exposed to and injected with the lunar material, showed no adverse alterations or ill effects from exposure. Since exhaustive studies of the astronauts and return lunar samples indicated there was no hazard to Earth's biosphere, the Interagency Committee on Back Contamination in January of 1970 concurred in NASA's recommendation that stringent quarantine rules be abandoned for future Apollo missions to the moon. Again, NASA reference there, of course. As I have mentioned, although the formal quarantine for the crew, spacecraft, and lunar samples was over, after Apollo 14, procedures for handling lunar material and protecting it from contamination remain in effect for the Apollo 15, 16, and 17 missions. For missions 15, 16, and 17, I moved my focus a bit to also include macrophage studies after lunar soil exposure. The macrophage is an important component of the innate immune system that plays a strong regulatory role as a vital link with immunity. The procedures involved peritoneal lavages, washes of mice and other species after lunar injections and incubation. I also conducted extensive still movie, cine or movie, film, photomicrography, and macrophage behavior, ingestion and reaction to the soil particulates. This exposure to the documentation aspects of the research is probably the main reason that I later moved into medical scientific media, and even much later, just into media. And today I'm an artist and photographer. To the right, another great photograph. Jim Irwin, lunar module pilot, pushes the core tube in by hand before hammering it the rest of the way. Charlie Duke, to the left, Charlie Duke, lunar module pilot, made this photograph of the core tube after hammering it into the ground, Apollo 16. And to the right, Gene Cernan, commander, made this photograph of the core tube after hammering it into the ground, Apollo 17. So I can't be exact which core tube samples I worked with for these missions, but I indeed worked with the lunar soil taken from core tube sampling. I refined the peritoneal lavage technique at the Lunar Receiving Laboratory and published a paper in Laboratory Animal Science. Later, when I was a research associate at the University of Texas Medical Branch at Galveston, 75, it's also a long time ago. Wow. I have one huge regret. I had a great deal of memorabilia from that period of time. And over the years and moving and here and there and to and from, unfortunately, I have lost all of that memorabilia. That's one of my big regrets in life. But anyway, to the left, you'll notice the Lrl mission badges that we wore. And for each mission, you'd have your name and mission number. This one is generic. Sorry. But I wore this kind of badge each and every day for 3 years. I wanted to mention Phil. He is seen to the right. I can't even remember his last name, unfortunately. It's terrible. This photograph shows Phil. He was a research analyst in botany, working with plants in the containment cabinet, just a couple of doors away from my lab. Our labs were, as I mentioned, were close in proximity. You'll notice his boot that we all wore. So I basically looked this way as well for 3 years. Or sorry, no, I take that back. Through the 11, 12, and 14, I think we relaxed in our street clothes for the other missions. Phil was small in stature, and he was frequently called over to the spacesuit training area to put on and test the suit for functionality and maneuverability, as you see to the right. He asked me to go with him on a couple of occasions to take photographs of him and the process. I did this and then handed him the camera. Man, I wish I had those photographs today. On my return to the Lunar Receiving Laboratory, Building 37 in 2016, the interior configuration had changed so much that I really couldn't find my way, or should say old way. I eventually figured out the location of my laboratories with the help of a couple of people, crew reception area, the spacecraft area, which at that time they had converted into a snack bar. I also made it over to the Lunar Sample Laboratory Facility, Building 31, where Andrea Mosey, lab manager, showed me one of the lunar rocks. You can see here. This is Building 37, back in 2016. Thank you very much. Allow me to get out of this presentation. I would like to show one more thing, and then I'll be open for questions, so bear with me a moment. Let me end the show. Sorry, just a moment. Well, I'm not doing a very good job here, just a moment. Okay. I'm back. That's okay, I'm back. So I hope everyone enjoyed that.
Yes.
Okay.
It was.
Okay.
Are you ready for questions?
I think I'm ready. Yes, ma'am.
Okay. So I'm just going to remind everybody. One, we've got lots in the chat, so I'm about to go over those. But if you have a question, you can unmute yourself. There's a raise hand function at the bottom, or you can actually like physically raise your hand, Annie, and I'll keep an eye on the crowd. And if you do either of those and we can't hear you, just make sure you unmute yourself or I will unmute you. I feel like and I wrote down so many questions, and then I think I feel like we could have a whole conversation about just what you could teach us about like not contaminating during times of COVID. So I feel
I
like we
hope I
could,
can answer these questions. I don't know. We'll see. After 50 years, I don't know.
we could go so many ways. So I'm going to start with some of the ones from chat. And then again, folks, if you have questions, please feel free. And if you also want to be able to like ask in person, just make sure you turn on your video. And again, unmute yourself when it's your time. Our first question is from Maury, who's one of our regulars. And I actually wondered about this too at the beginning. When you were talking about the mice that are like completely isolated and have no germs or any of this stuff, how can a mouse live without bacteria in their stomach?
I think they probably have beneficial gut bacteria. We're talking about pathogenic, maybe,
Okay.
pathogens. So, yeah, I think all animals, again, all animals have beneficial gut bacteria.
And then I'm going to stay sort of in the veins of stuff. So folks, if you have a question, I'll go in themes and I will get to them all, hopefully. Related to the mice, two sort of follow-up questions. What would have happened if one of the mice had tested positive for contaminants from the moon?
Continued isolation, continued quarantine, and the powers that be would have stepped in, not me. I was a lowly research analyst, if you will, and way down on the totem pole. But the powers that be would have stepped in and would have made their decisions. I don't know what those decisions would have been, but certainly a part of that decision would have been to continue to isolate and quarantine that mouse in that area until things could be figured out.
Also, because you were still chatting with folks from NASA, and obviously you were just back in the last few years, do you know if they now use alternatives to animal testing, or do they still use the same methodology?
I have absolutely no idea because I am totally detached from that.
Okay.
My only contact with any NASA type, particularly at the LRL, was in 2016. And that was, just 4 years ago. So I really have no idea what kinds of I know medical. In fact, I think a person is here, Judith Hayes, biomedical group. She might be able to answer that question, but I have no idea about their testing procedures and how they go about their activities today.
Yes, I'm here. I don't know if you can hear me.
We can hear you.
Yeah, thanks for the invite and the great presentation. It's always good to hear the historical perspective of Building 37 and the Lunar Receiving Lab. And yeah, there's some alternatives that we use. We still use a lot of animal research. We do it every day on the International Space Station, so it's still a big part of research at NASA. At least the rodent research is. But there are some alternatives with bioreactors and other tissue models. But, animal research is still out there. And there's been some advances in some of the animal work as well. And unfortunately, we're no longer in Building 37. We vacated that 2 years ago, and we now have a new human health and performance laboratory. And they're planning on tearing Building 37 down, which is unfortunate.
Shameful.
Yeah, it's a historical facility, and I hate to see it go, but that's on the plan to be torn down.
If I may, Rachel, I'd like to ask Judy a question. Judith, did you know a Rudy Landry?
I did. I put in the. I put in the chat what a good man he was, and he was a fine scientist, and he did continue to work in Building 37 in the laboratories and the clinical labs and the immunology lab for years. He was a wonderful man.
Well, we were best of buddies from 69 to 72, 73, the best of buddies. And his wife, it's been several months ago now, but sent me an email and said that he had passed away. So that truly, truly hurt me.
Yeah, yeah, I'm sorry to hear that too. He was a good guy and a great scientist.
Yeah.
Thank you so much. And thank you for, like, it's so amazing to have someone who's still affiliated who can answer some of those questions. So it's lovely to have Tom, and it's lovely to have you be able to give that
Yeah, there's a
perspective,
couple of us
Judy.
here because we're fond of this topic, so we wanted to hear his perspective. So thank you for allowing us to sneak in.
I, the next question is like the questions that people want to ask, but not everybody will ask. But again, our good friend Maury will ask, which is, okay, so you talked about you had to get in this suit, and then you also had to take these like decontamination showers, etc. Did you get like bathroom breaks in this bunny suit? Like, how did that work? Or were you just in there for 8 hours with no breaks?
Yeah, we had toilets in our area, in our immediate area. So once we put on our bunny suit, kind of very similar to a surgical uniform, but once we put on our bunny suit and walk through the UV wash, dry wash, and into our area, there are bathrooms and all of that. So no problem there. No problem. Yeah.
I did love this question. Annie wanted to know what were the most surprising results that came from your testing? Like, what would be your main takeaway from that period?
Well, surprising. I think all of us down deep, because all of us had, even though I was very young and just out of college, but I did pretty well in college, and I actually had a research position at MD Anderson Hospital for a couple of months. We're in fact working in microbiology. I think all of us were trained and had this feeling that we weren't going to find anything, because most test subjects, if you will, say animals, plants, treated with soils from all over the earth, it's pretty inert. There are not going to be adverse reactions. So we had a similar feeling that we're not going to see anything big here. It was, so, and indeed it was inert. I take that back. In botany, and I wish Phil was here too. I hope Phil is still around. Judy, if you're still here, did you meet a Phil in botany?
No, by the time I arrived, we
Yeah,
weren't
okay.
doing the botany work anymore.
Yeah, I think he's probably long gone. But I believe Phil mentioned to me, and you can certainly research this on the internet, there were at times a few spurts of growth in some plant species because of the lunar soil treatment. I'm not sure what that means exactly. I'm not a botanist, but let's say it was more positive than negative. So we, in our section, biological, zoology section, if I put it that way, there were no adverse effects. And we really didn't anticipate that in the first place. But it was a good idea to go through that process because you never know.
I had a question, and we still have many more to get, and we're going to try to get to them all. But one of the things that really struck me is you talked about your first night there and this sort of numbness after this first shift too, like, because I think of the heightened, how careful you were being because of contamination. I don't want to use the word fear, but you were being, really diligent. Did that become more regular? Did you always sort of feel that way, or did it become something you were much more comfortable with, or did it always sort of have that heightened,
Yeah, that's
again,
that is a great question. I've never been asked that question exactly that way. First of all, please keep in mind I was only 22. I just turned 22. So I was very young and impressionable and scared, maybe, I don't know. But it was, this was such a unique situation for me, and a different situation for a youngster. Even though I was degreed in biology with some experience, walking into that situation, and particularly at night, a night shift. When you think of a night shift, it tends to skew your perspective a bit. Night shift, that's a little bit mysterious maybe, but having to walk in at night, dress up in a surgical outfit, no masks, by the way, but surgical outfit, being led by the hand by your team leader, Landrum. Judith, did you know a Landrum Young?
No, I didn't. I don't know if John Urie, I think, is online too. I don't know if he knows them either.
I'm sorry, I'm trying to find these people.
Them personally.
Sorry? I heard somebody.
Yeah, Judy just mentioned my name. This is John Urie, also from JSC. I know the names, and I actually tried to look up pictures of Phil in our database, but he was never identified by name, so I can't help you with the last name, unfortunately.
Landrum Young, did you know Landrum Young?
I did not know him either, only by name and reputation.
He was my immediate boss, my team leader. Actually, we communicated for a few years, and now it's been several years since we have done that through email, and I'm afraid that has passed away. I just don't know. Rachel, sorry, back to your, anyway, Landrum led me by the hand, so to speak, through the UV wash, dry wash, walking down this you saw a picture of it, this very long corridor with these autoclaves, like they look like you're in a submarine with the hatches. It was just so surreal. It was so different for me. Walking down this long hallway to the very end, having to punch in numbers to get into the laboratory. Top secret, it seemed like. So it was just so different for me and so surreal. And particularly when he opened the door and I saw the containment cabinets for the first time. It was a bit of a sci-fi moment, if you will. A bit of a sci-fi moment. And for me, it was reality and surreal at the same time. And I'll never forget that. You also mentioned, did it wear off? Working with the lunar soil never wore off. Seeing the lunar soil for the first time, actually I got in at the tail end of Apollo 11, so the lunar soil was already absorbed, so to speak, within the animal species. So I really never ever saw Apollo 11 lunar soil in the containment cabinet. But Apollo 12, I was right there at the beginning of that mission. As soon as I think my team leader, Landrum, took the lid off the metallic tube and we peered in, that was a eureka moment for me. Unbelievable, unbelievable. To see the moon just inches away from my face on the other side of the containment cabinet. I'll never forget that. Same for Apollo 14. Same for the other missions. Seeing that lunar soil, the moon, that close, not being able to touch it, but being that close to the moon was always a wonderful moment. In terms of the procedures and having to walk down the corridors and put on the bunny suit day after day after day, that got pretty routine, yes. But the lunar soil and that first night is imprinted in my memory, yeah.
I think I can speak for everyone here. I feel really honored to be able to hear that from somebody. It's just it's so neat it like makes me think about it in a totally different way. And so I really appreciate your perspective. This is actually somewhat related just because you were talking about the I don't want to call it soil, but I guess the soil samples from the moon.
Well, it's called lunar fines, F-I-N-E-S. So it's very particulates anyway, yeah, sorry.
Good to know that will be what I learned. One of my main learning pieces for today. There was a good question there from Kenneth. Were there any problems with the design of the core tube?
Oh, I can't answer that because I'm not exactly? I'm not an engineer, and any problems, I have no idea. That's between the engineers and the astronauts and the technicians on that end. I'm sorry, I can't answer that. And I didn't work directly with the lunar rocks and the core sample tubes. That was all done in the geology section. By the time we got to our sample, as I mentioned in the presentation, our sample had been filtered, sealed, prepared, and put in a container for us to work with. So that is as close as I got geologically to the moon. I'm totally apart from the geology laboratories, and we never crossed paths. Again, I mentioned we were so compartmentalized, we never saw each other. And we were also very, very busy. Yes, there was moments of enlightenment, if you will, and joy and all of that, but our jobs were so tedious and so exact and laborious at times. That's all we thought about 99% of the time. And I had a job to do. I went in, did my job, and I left and went home. It's pretty much how it was.
We're going to take 2 more questions, and then just a little bit of follow-up information. One, and I think probably a few of us are curious about this, and I wrote this down too. You know, you were at MD Anderson, and then this person was contaminated, and you kind of got this amazing opportunity. What is your degree in, and where did you attend school?
Well, my degree's in biology. I have a BS, Bachelor of Science in Biology from Midwestern State University in Wichita Falls, Texas. And I had worked on my master's at one point in time, and I'll leave it at that. I moved on, did other things. So anyway.
Understood. And then our last question is from Annie. What did you think of the 1971 film, The Andromeda Strain?
I can't even remember watching that movie. I haven't even seen a rerun of that. No, I'm being facetious. Very, very similar procedurally and technically, and very, very similar. Outcomes different. Speaking of that, I don't mind mentioning this. What we did 50 years ago, I think we should be doing today with COVID-19. I'm sure that CDC is doing this. I mean, there are areas all over the world that are doing what I did 50 years ago, probably. But I wish we would take as much care privately, personally, in groups. We should avoid groups, but I think we should take as much care as we did 50 years ago with the lunar sample.
Did he freeze? I think he might have frozen. Okay, we're gonna hopefully he pops back, but we're near the end of our program. So, oh, there we go.
Yeah, you froze.
You froze for a second, but I think we're good. I'm gonna do a wrap up, and then, well, actually, this is really truly our last question, but I also kind of was curious. So we just talked about your degrees. How did you get the opportunity to go from MD Anderson to analyzing lunar soil samples? Like, how did that happen?
I think I mentioned that a bit, but yeah, it's truly interesting. I had no idea, as I mentioned on my very first slide, watching Armstrong and Aldrin walk on the moon with my grandmother and my parents on July 20th, I had absolutely no idea. I didn't even have a job at the time. I had just graduated 2 months earlier from school, from college. I didn't even have a job. So I certainly had no idea that I would be working at the Lunar Receiving Laboratory come September of that year. So at MD Anderson, working in the microbiology area, things were okay, but as we all do, we look for other positions to check out things and see what might be better, so to speak, or maybe more apropos for who you are and what you want to do. So I happened to see an ad in the Houston Chronicle. They're looking for a research analyst at NASA, Houston, the Lunar Receiving Laboratory. I answered the ad. And I don't mind saying this, sometimes it's who you know, right? It's who you know. The team leader, Landrum Young, who I've mentioned more than once, he had worked at the same laboratory at MD Anderson that I had worked, or I was working at. So he called me and he said, Tom, I'd like to meet with you because I used to work where you're working. So there was a bit of a connection there at the very beginning. I said, great, I would love to come out and talk with you. So I drove out to Clear Lake City, which is south of Houston. I drove out to Clear Lake, and it wasn't much of an interview because Landrum hired me almost immediately because we were working buddies, so to speak, even at a distance, at different time periods. Of course, he looked at my GPA, and it was not too bad. I had A's in all of my biology classes. And but that, along with my degree and my grades, particularly in biology area, and the fact that we had worked not only in the same area, but for the same MD, PhD type, the person, he said, Tom, I'd like for you to come to work. I said, hey, it would be great. It would be wonderful. And I accepted that position, well, knowing, of course, about the Apollo program. And, you know, I'm a Mercury, Gemini, Apollo baby, kid. I grew up with all these missions. So, of course, I knew about all of this, but I really didn't have the details and what I was about to get involved in when I accepted that position. And that takes me back to that first night that was so surreal for me.
We're going to stop there because I think it's kind of perfect ending. And I also want to be really respectful of people's time, but I think it's a fascinating story. A couple of things, just a reminder. So we don't like spam anybody, and I won't add anybody to any email lists or anything like that, but I'll email just the group that was here that registered a link to the video once we have it up on our YouTube channel. And Tom graciously had his email listed in his PowerPoint. So as long as he's okay with me sharing that as well, we can put his contact information in case you have further questions, or you can always email them to us, myself or Annie at Shalom Austin, and we can make sure we get them to Tom if you had a question that didn't get answered or you didn't have time to ask. We just want to thank you all so much for taking the time and attending, but really the biggest thanks to Tom for volunteering his time. We're so lucky to have him in our community, and so cool to be able to have him here and speaking. And so just a really big thanks to Tom. And then it was wonderful to meet many new faces in this program.
So thank you, Rachel, very much. It's just great to be able to reminisce this way. So the older I get, the more I miss the past, you know, so this is great. Thank you very much.
Thank you, everyone. Have a wonderful rest of your day, and we hope to see many of you at Shalom Austin programs in the future. And everyone stay safe and healthy.
Okay.
Bye-bye.